It's my pleasure. Thank you very much for this invitation. I'm very happy to talk at the seminar series of the Ecological Forecasting Initiative. And today I will be talking about quantifying resilience of populations and unraveling mechanisms behind it. And for some reason, I'm stuck. Maybe now it's okay. Now it looks like it's working. So in the world that experiences diverse types of disturbances of increasing magnitude, it's important to assess their impacts on populations. Classically, in demographic studies, to assess such impacts, we focused on population viability, and we rarely spoke of resilience. That is why in my talk, in the first part, I will first make a little excursion into population viability analysis, a tool that is used to assess population viability. Population viability analysis mainly focuses on the long-term asynthetic population dynamics. Importantly, in response to a disturbance, the population may also exhibit short-term dynamics, and this dynamics is often now recently, since 2022, is quantified using the concept of demographic resilience. But it has been also quantified before using transient analysis. So in the first part of my talk, I will be talking about short-term population dynamics in response to perturbations and long-term population dynamics and the tools used to quantify these behaviors. Quantifying resilience, of course, is an important step, but it's just one step in diagnosing the state of population. As the next step, it's very important to try and understand the mechanisms that render a population resilient. And in the second part of my talk, I will focus on one such mechanism, phenotypic plasticity. So population viability analysis is a well-established tool that uses models to assess viability of a population under a range of plausible scenarios. For example, climate change, habitat fragmentation, and restoration management. There is no one single model that is used for PVA. They may differ in their structure and complexity and the level to which they incorporate processes. However, usually as input PVA model requires at minimum the biological data, so the data that describes the life cycle of the species. And of course, if it's a spatially explicit model, we may also use landscape data. Then what we typically do, we project the population size over time, and we often use stochastic models. So therefore, you see different simulation runs resulting in slightly different outputs. And in one of these runs, depicted in orange, the population goes extinct. As output of PVA model, we obtain estimates of population viability, for example, extinction risk and metapopulation size. And as I said, there are different types that can be used for population viability analysis. So one could use unstructured models, such as ordinary differential equations, or patch occupancy models that rather focus on the metapopulation dynamics and emigration and immigration processes, and not so much on the demographic dynamics within a population. Another tool that is arguably most often used in population viability analysis is matrix population models. They structure the population in stage or age classes that are characterized by different survival and fecundity. And commonly, survival and fecundity are referred to as demographic rates or vital rates. And another type of the model that is probably the most complex are individual-based models that describe the fate of each single individual in a population, and the behavior as well as survival and fecundity of these individuals is then a function of environment. And then as a sum of the fates of each single individual, the overall population dynamics emerges. So since matrix population models were so instrumental and were so often used in PVA, I would like to give you a brief example of the application of the use of matrix population models in a PVA. And I would like to use the study from my PhD thesis that I have conducted a while ago, in which I focused on assessing viability of a bog fritillary butterfly in Ardennes region in Belgium. This butterfly species is called a glacial relict species, which means that it inhabits the habitats that were left by glacial period. These habitats in Western Europe are peat bogs and wet meadows, and they are separated by hundreds of kilometers of unsuitable habitat from the larger habitats that are available again in Scandinavia. Since this species is a glacial relict species, it favors colder and wetter conditions, which are more threatened by climate change. And therefore, it's very important to assess the viability of population in face of climate change. So to assess the viability of the population under climate change, we had to assess the vital rates of each life stage. Briefly about the life cycle of this species, there are adults, they lay eggs. From these eggs, with a certain probability, little tiny pre-diapause larvae hatch. Then they feed until autumn when they go into a diapause, and then in spring they wake up from the diapause and feed a lot again. We call this stage post-diapause larvae, and then they pupate, and eventually, with a certain probability from these pupates, new beautiful butterflies again emerge. So in order to quantify the vital rates to be used for our population matrix models, we conducted field work, part of which was macro-capture study to assess the current population size of this population. But we also collected some female adults and put them in the cages in the lab to lay eggs. We then took those eggs and submitted them to 3 different temperatures in climatic chambers. And similarly, we then exposed larvae and pupae that survived from those initial egg batches and exposed them again to 3 different temperature treatments. We also conducted semi-natural experiments whereby we placed the first instar larvae, so these pre-diapause larvae, in the cages that you can see here in the field. And then in the next summer, we looked at the rate of emergence of butterflies. So these experiments allowed us to parameterize the population matrix models with the survival rates and fecundity of different life stages under different temperature treatments. Matrix population model shows the probability of transition from the stage in the column to the stage that is shown in the row. And for some reason, I see that my pointer is frozen, but I hope you see the idea. So 0.75 value here shows the 75 proportion of eggs that turn into pre-diapause larvae. And then 89 in the first row in the last column shows that the per capita fecundity of butterflies is 89 eggs. We constructed such different matrices for different temperature conditions, and that allowed us to assess long-term asymptotic population growth rate. So on a baseline scenario, we found the population growth rate was 0.97, which is less than 1, and so is typically interpreted as a decline in population. Under the very high, which is a larger unlikely temperature scenario for Europe within this century, the population would decline with the growth rate of 0.56 and the associated time to extinction of 12 years. Our sensitivity analysis also indicated that survival of pre-diapause larvae is critical for the population viability of this species. So to prevent the population decline, we suggested establishment and support of tussocks of these grass structures that you see here. And in order to allow these tussocks to establish, we suggested removal of the trees and bushes in a close vicinity of the peat bog to increase the groundwater table. So PVAs often use asymptotic population growth rates, as I just demonstrated in the case study, to make conclusions about population viability. Importantly, asymptotic population growth rates are expected to be reached by a population in constant environment. However, this is rarely the case in nature. Disturbances often push populations away from a so-called quasi-equilibrium. So according in this exemplary plot here on the right hand, according to the asymptotic population growth rate, which is 0.95, this population will decline on the long term. However, its behavior in a short term, in response to a perturbation to a demographic structure, may in fact deviate strongly from such a long-term behavior. As you see on the figure, right after disturbance, the population density may decline compared to the expected population density under asymptotic behavior, and this we call attenuation. And then it can overshoot, it can be higher than the population density expected under the long-term population growth rate, which we call amplification. The period over which the behavior of the population deviates from the one expected under asymptotic growth rate is called transient periods, and the respective population dynamics is called transient dynamics. So at a certain point in time, and I'm not sure whether you see my pointer, I'm looking at the, yeah, okay. So at a certain point of time, you can see here that the solid line that shows the observed behavior will be parallel to the dashed line, which means that the growth rate of the observed population, of the disturbed population, will reach the one that is expected under asymptotic behavior. And that's where transient period ends. So such transient dynamics can be studied with transient analysis that allow measuring the short-term response of populations to disturbances. And in fact, transient analysis have been used in demographic studies, however, arguably rather rarely. So recently, Paul Capdevila and colleagues called for a more widespread use of transient analysis in population ecology. And to do that, they introduced the concept of demographic resilience and the framework for quantifying it. This framework relies on the application of transient analysis to matrix population models in order to derive a set of resilience metrics. And this study was inspired by their finding that ecological resilience so far have been mainly studied at the community and ecology, in community ecology and in ecosystem ecology, and it was much less investigated in the field of population ecology. So the concept of demographic resilience, as proposed by Capdevila and colleagues, derives from classical ecological resilience. And there are clear parallels in how resilience is quantified at these different levels of organization, community, and population. On the one hand, the abundance of each life stage or age classes in a population, which we call a population structure, here shown on the right-hand side, can be compared to abundances of different species in a community, which is called community structure. Another parallel, another similarity is that in a community matrix model, each cell reflects the effect of a species, of the abundance of a species in a column on a species in a row. And the diagonal shows the effect of the abundance of this species on itself. So that's what we usually call density dependence. Similarly, in population matrix model, as I already have mentioned, the cell reflects the transition probabilities from a stage or age class in the column to the one in the row. Demographic resilience is defined as the ability of populations to resist and recover from alterations in their demographic structure. Thus, demographic resilience, just as ecological resilience more broadly, sensu lato, has a so-called bivariate character and requires measuring two components, resistance and recovery, where resistance is defined as the extent to which the population size is affected by a disturbance, and recovery is the time that is needed for a population to come back to its pre-disturbed or quasi-stationary state. So this set of metrics belong to those that actually consider one single equilibrium for a system. And in fact, it's not only two aspects, resistance and recovery, that can be measured, but there is a suite of metrics that can be used to measure those two aspects. And I will highlight some of them, exemplify some of them on this schematic figure, where we have population size changing over time. And for the simplicity, we assume that the long-term population behavior would be a stable population dynamic. So that's shown by this dashed line here. However, a disturbance may push population away from such a behavior, and it can exhibit, for example, the pattern shown on the slide. We can measure the following resilience metrics. Reactivity, which is a decline in population density at the first time step compared to the density that would be observed under a long-term asymptotic growth rate. So here, for simplicity, it's a stable, it's a parallel line. We can also measure maximum attenuation, which is the lowest population density that can be reached at any time step after a disturbance. And for populations, unlike for communities, a disturbance can lead to overcompensatory dynamics, which would result in increasing abundance after a disturbance. And this is captured by maximum amplification, which is analogous to maximum attenuation, only that it now measures the increase in density after the disturbance. We can also measure inertia, which is a long-term offset of a population density after the long-term population growth rate is reached from the population density that is expected under asymptotic behavior. And finally, dumping ratio is a measure of convergence to stable growth and reflects recovery time. So the current framework for quantifying demographic resilience suggests that if demographic rates were measured in multiple years, they should be averaged over time, resulting in a so-called time-averaged population matrix. This returns one single value per resilience metric for the whole time period. However, a population can experience disturbances of different kind and different intensities over time. And we expect that such disturbances will cause demographic resilience to also vary over time. To assess such time-varying demographic resilience, we propose to freeze the demographic and environmental conditions at each time step and use them to assess what would be demographic resilience if such conditions had persisted till population recovery. Thus, by increasing the temporal resolution of analysis, we try to phenomenologically unravel the time points at which demographic resilience was especially low or especially high. And then the time points, years, for example, with very low resilience would point out, hopefully, to the likely disturbances that affected the population. And we study this time-varying approach, and we study to what degree and in what conditions time-invariant approach still holds within the project that is called Wilder, that was funded by the BMWF, and that is co-led by me, Sarah Benheim, and Oliver Höhner. Sarah and Oliver are the leaders of two long-term studies on spotted hyena populations in Tanzania. And I will speak about those populations just in a moment. So we demonstrate the relevance of the time-varying approach in a case study using the rich demographic data on two populations of spotted hyenas. These data are coming from Serengeti National Park, that's one population, and Ngorongoro Conservation Area. Both of these populations were monitored for more than 25 years, and so represent a very rich demographic data, also the very high temporal resolution. Briefly on the hyena biology, these are social group-living carnivores with a female-dominated hierarchy. For them, social rank determines access to resources and therefore is very important for demography as well. And the female gives birth to 1 to 2 cubs per time. So we used these two long-term studies on hyenas to address two research questions. First, how well can time constant approach approximate resilience and for which metrics? And second, what is the impact of initial population structure on calculated resilience metrics? And this work was conducted by Ella White, a PhD within Wilder. So I'll try to explain her work as best I can. And so regarding the second question about initial population structure, I already explained to you the population structure is the proportion of individuals in each stage or age class in the population. And commonly, in terms of the initial population structure, the current framework to quantify demographic resilience uses stage-biased vectors. And so what are stage-biased vectors? Those are the population vectors that basically are presented by the populations in one stage only. That's why they are called stage-biased. So on this figure here, we have all individuals are cubs. So the whole population is in cub age class. Even though the use of such stage-based vector is very useful for comparative studies, it was unclear to us whether they are as relevant also for conservation. Therefore, we also investigated empirically informed population structures, which are constructed by using the number of individuals in each stage class from the field data. And I must say that this is possible in this luxurious situation where we have very detailed information on this population, but very often such cases will not be available because the data usually for PVA that is collected, if you talk about PVA, are not of this kind of resolution. So we also used what we call randomized structures, where we drew 1,000 random population structures from Dirichlet distribution. But for the sake of staying short, I will just focus on the two stage-biased and empirically informed one for this presentation. So briefly about the framework that Ella used. For the matrix population model, she used the life cycle that distinguish demographic and social states. We distinguish the following demographic states: CAPS, sub-adults, and they are here abbreviated as C, as A, etc. So we have sub-adults, young adults, and then we have adults that can become breeders, that can be breeders or non-breeders depending on the year. The temporal resolution of this matrix is yearly. And then each of these states could be in high or low social state to capture the strong impact of sociality on demography, particularly on the survival. Ella then used multi-state macro capture models to assess annual survival rates for each life stage. As a next step, she combined these annual survival rate estimates together with the fecundity estimates that she obtained separately for each year and parameterized annual population matrix models. They are shown here in cyan or green. And from each of these annual matrix models, she could then derive, using the transient analysis, a suite of the resilience metrics that I have just outlined before. We then also done what is commonly done currently. So we averaged over the vital rates over time to obtain a single time averaged matrix, which is shown in violet. And then we also use this one to calculate the resilience metrics under a so-called time constant approach. And finally, we assessed the discrepancy between both approaches by using the mean absolute percentage error. And so this metric ranges from 0 to 1, with 0 meaning that our time variant estimates here would all lay on the violet line. So it would mean that time constant approach is sufficiently well capturing the sufficiently well approximation for reflecting the dynamics of the population. Whereas if MAPE reaches closer to 1, it would mean that the variation over time is so important, time matters and has to be included, has to be considered in the analysis. And so we used a threshold of 0.05 to say whether time matters or not for quantifying demographic resilience. And first, I would like to show you the result using the stage-biased initial population vectors, so something that is commonly done currently with the framework. And you can see here this MAPE measure, so it's the measure of the discrepancy between both approaches for each population and for each matrix. And as you can see, apart from this one value here, basically all of them are higher than 0.05, which means that time matters when quantifying demographic resilience. And it's especially important, the discrepancy between both approaches is especially high for maximum attenuation and lower inertia. We then also calculated demographic resilience metrics using the empirically observed initial population structures. And in this case, we got slightly different results. So we found, again, using MAPE as a threshold of the threshold of 0.05 was exceeded for inertia in both populations and for maximum attenuation in Serengeti population, meaning that not only time is important when quantifying resilience, but also the demographic resilience, the conclusions about demographic resilience vary if we use different initial population structures. We also asked the question of whether inclusion, this is in a different project, whether inclusion of time when quantifying demographic resilience is especially beneficial for certain life history strategies. And as a proxy for life history strategies, we used generation time, a life history trait, and so we assessed how generation time is related to the discrepancy between the time constant and time varying approach. And as a discrepancy, again, we used the same metric I have shown to you before, MAPE. This work was conducted by Malte Purok, a bachelor student who was supervised by Julie Louvrier and myself. And to answer this research question, we searched Comadre data set, and we extracted the studies that were at least 10 years long. So they provided population matrices for each year for at least 10 years. And we excluded human studies. As a result of these criteria, we were left with 19 studies on 14 species. Remarkably, majority of these studies are in the global south, specifically in Africa. This is, I say remarkably because this is rather unusual for this kind of global meta-analysis. However, on the other hand, of course, this result is linked to the fact that our dataset is very much biased to primates. So keeping in the head of your minds these limitations and biases of the dataset, these are the results that we found. So here on the y-axis, we have our measure of discrepancy between both approaches, MAPE, and on the x-axis, we have generation time. And what we found is that discrepancy between both approaches is highest for short-lived species, only for two metrics, reactivity and damping ratio. Importantly, we facilitate quantification of time varying resilience and comparison of time varying resilience to time constant approach by providing the R package, DEMRES. This package was developed by Julie Louvrier, the former postdoc in the lab, and we hope for its future use and application, and hopefully more insights on the time varying manner of demographic resilience, or more support for the time constant approach. And similarly, I would also like to highlight another R package that was developed in my group. It's E-STAR that was developed by Ludmila Figueiredo, a former postdoc. This package is broader in its application in that it allows quantifying stability at different levels of organization. And it provides two different sets of stability metrics. On the one hand, it provides metrics that can be applied for what we call univariate time series data, for example, abundance or species richness as a data. And then we can calculate some typical metrics as resistance and recovery. However, it also allows quantifying asymptotic stability metrics using the community matrices. So the take home from this first part is that classical PVA tools focus on long-term population dynamics. Demographic resilience framework allows quantifying population response to perturbation in demographic structures. Our research shows that time and population structure should be considered when quantifying demographic resilience of particular specific populations. We also found that time is especially important to consider when quantifying resilience of short-lived species, and we provide tools for quantifying resilience. So as I said already at the beginning, now I'm coming to the second part of my talk. And as I said at the beginning, quantifying resilience is just like a first step in diagnosing how well the population is doing. For a conservation of taking particular actions, a next step, and very important one, is to try and find out what are the mechanisms that render a population resilient. And in this part of the talk, I will focus on one such mechanism, phenotypic plasticity. Specifically, we look at the role of phenotypic traits in mediating animal population responses to climate. And before I start with it, I would like to acknowledge that this is a large collaborative effort, and this study would have not been possible if not for efforts of many people in collecting long-term population data, field data. And this study started by an SDIV synthesis grant at IDIV in Germany. So I would like to also acknowledge the synthesis group's trait change. So I don't think I have to convince anyone in this audience, even though I don't see you, but I hope very much I don't have to convince you that climate change is happening. And one of the characteristics of climate change is the increase in the long-term mean, which we call global warming. Climate change affects multiple species traits, among them phenology, which is the timing of the recurrent biological events such as reproduction or migration. Examples of such phenological traits are the day of the first arrival of migrants, rat date, and the mean flight date of butterflies. There is a strong evidence across species that phenology is advancing with warming temperatures. Another group of traits whose responses to climate are quite well studied are morphological traits, for example, body size and body mass. There are some studies showing that these traits are decreasing with warming temperatures. For example, the bills of red knots have shortened with warming temperatures and earlier snowmelt dates. However, for other species, an increase in body mass was recorded with warming temperatures. And a prominent example is yellow-bellied marmot. Individuals of this species started to emerge earlier from hibernation in response to warming temperatures, which led to longer growing seasons and respectively heavier individuals. Importantly, changes in climate affect not only phenotypic traits, but of course also populations. In response to warming temperatures, populations of some species have increased. So if we stay with the example of the yellow-bellied marmot, individuals with larger body masses had higher survival rates, which led to a very quick, abrupt increase in the population size over the last 2 decades. However, not all species, of course, benefit from climate change. Some populations decline or go extinct altogether, as it happened to large blue butterfly in the end of the last century in the UK. So, although both phenotypic traits and population numbers are affected by climate, classically population ecologists focused mainly on how population sizes are affected by changing environments, for example, climate. So the focus of population ecologists was on the effects of environment on population size or on demographic rates, whereas phenotypic traits were not so much of a focus. On contrary, evolutionary biologists study the effects of environment on trait and fitness consequences of such trait changes. There is thus a deep divide between these two disciplines. However, incorporation of the traits in population ecology may allow us to better understand the mechanisms via which populations respond to environmental changes. Indeed, if individuals respond to environmental change by changing their traits, be it behaviorally or morphologically, that may ensure population stability. Thus, by studying such mechanisms as phenotypic plasticity and to what extent it contributes to population stability, we may get deeper insight into how populations cope with environmental changes. And so, in line with this, in this research, we asked the question, to what extent do phenotypic traits mediate population responses to climate across animals globally? And in a very simplified framework that we used, we are mainly interested in this path shown by the solid black line. So we are interested in the trait-mediated effect of climate on population growth rate. And we do also account for all other effects that do not go via this trait, but potentially via other traits, or even the direct effects of climate on demography with the dashed line, with the arrow shown with the dashed line. So to answer this research question, we conducted a systematic literature review, looking for the studies that recorded phenotypic trait and population size over time, with the minimum study duration of 10 years, and we searched across animals worldwide. And I would like to acknowledge Karis Jones, an intern at that time in the team, that made a tremendous work in assembling the data that was coming from this systematic literature review. Without her support, I think this wouldn't have been possible. So our literature review returned more than 1,000 abstracts, from which 73 papers were retained as satisfying the criteria. And that resulted in 213 studies on 74 species. And so we have these studies split into two trait categories, phenological and morphological. This is the spread of our studies geographically. Unfortunately, as is the case with this kind of meta-analysis, we have a strong bias towards Northern Hemisphere and towards birds. We are speaking of long-term studies, so the median duration of phenological studies is 25 years, and the median duration of morphological studies is 14.5 years. We focused on the effects of two climate variables, temperature and precipitation. And for each study, we extracted the temperature and precipitation that best predicted the trait using sliding window analysis. In brief, how we did this, we inspected the windows of all possible durations up to 2 years prior to the focal event. In this case, this is the parturition of a raw deer. We then used the mean for the temperature and the sum for the precipitation over the identified windows and selected the best one using the delta ASC to the null model. And for the sake of staying short, I will focus on our findings for phenological responses to temperature only in the remainder of this talk. So here a bit more detailed path diagram that we used in our structural equation model. What we are mainly interested in is this black solid line arrow, as already explained before. So the data that we use are time series of phenological traits and population growth rates taken annually, and the temperature variable that we found using our sliding window analysis. And these arrows basically here see that they are slopes of the effects of temperature on phenology for each particular study that we have in our dataset. And then ZG is the effect of phenology on population growth rate while accounting for temperature and population size, it should say. And we also account for population size to account for the widespread density dependence found in natural populations. And we did account for time prior to the analysis by detrending our climatic variable before fitting SEM. So these are the results directly. Here we see the relation between temperature and phenology, and each gray line reflects each single study. So we can see high heterogeneity in the responses of phenology to temperature across the studies. There are studies that show an advancement in response to temperature, but also there are those that show delay in response to temperature. However, across the studies, the phenology is significantly advancing with temperature, as shown by the thick black line here. Now we look for the next relation. So this is the effect of phenology on population growth rate while accounting for temperature. Again, there is a high heterogeneity among single studies, and across study effect, global effect is non-significant and is very slight positive effect. Importantly, because we are interested in this trait-mediated effect of climate on population growth rate, so we are basically interested in the product of both slopes CZ and ZG, and it's important to consider both slopes together. So now we look at how effects of temperature and on phenology for each particular study relate to the effects of phenology on growth rate. So in this plot now, each point shows on the x-axis the slope from the panel that you have seen on the slide before. So x-axis is the effect of temperature on phenology, and y-axis show the slope from the panel that you have seen on the right before. So the effect of phenology on population growth rate. So basically, to help you get your head around that, I highlighted one study in red and hope it's not too confusing that now we are talking about slopes being plotted on the x and y axis for each study. So each study each point is still a study, but the axis reflect the slopes from the two previous plots. Okay, you might ask why we do that. That's because it helps us to formulate our expectations. We expect that if phenology-mediated effects of temperature on population growth rate contribute to population dynamics, then points should fall into the quadrants highlighted in cyan here. We again talk about CZG as a product of these two slopes, and I highlight that we include all the CZG is supposed to be non-negative. So we do not only look at the positive effects, but also of a CZG being 0. We do not consider that only positive trait-mediated effects of climate on population dynamics are beneficial, because the trait can change in response to changing climates such that population numbers will be kept stable. So there will be no effect of trait on population growth rate, and therefore CZG would be 0. And these are the results. So we found that large proportion of the studies, namely 60%, fall in the two quadrants that were highlighted in cyan before, as expected. this proportion is significantly different from random, from 0.5, which we interpret as, on average, phenology mediates temperature effects on populations across the studied species in our dataset. Importantly, in large proportions of studies, ZG, so this y on the y-axis, is very close to 0, which is an indication that majority of these responses are likely due to phenotypic plasticity and not microevolution. However, this is just an indication, and of course, unfortunately, we do not have the detailed necessary data for each study to confirm that. We also attempted to explain the high heterogeneity in the effects of temperature on phenology with such species characteristics like diet, generation time, and latitude. Unfortunately, none of the species characteristics were able to explain this high heterogeneity. However, we found a negative effect of latitude on the association between phenology and temperature, such that the especially sensitive populations to temperature were found at higher latitudes. Yet the variation in our studies could not be explained by either ZG or CZG. We couldn't, sorry, no, I'm saying n'importe quoi. We could not explain the variation in CZG or ZG using the latitude. However, the variation in CG, so the effect of climate on population growth rate that is beyond the effect that goes via the phenology, was explained again by latitude, such that the direct effect of climate on population growth rate was most negative at lower latitudes, and it became close to 0 at higher latitudes. One possible explanation for both significant latitude effects is that the populations at lower latitudes have traits that are less plastic and hence have lower thermal tolerance than higher latitude populations. And so to conclude, phenological responses to temperature contributed to population dynamics. We found high heterogeneity in phenological responses to temperature. Latitude could explain phenological sensitivity to temperature and also the effect of temperature on population growth rate beyond the one mediated by phenology. We did not find species characteristics that could explain variation in climate responses to temperature, in phenological responses to temperature. Importantly, we found quite high intraspecific variation. It was as high actually as interspecific variation, and thus predictors that reflect local environmental variation, for example, habitat, may be more useful than such predictors as species characteristics. And to give you a bit of an outlook, in one part of my future research, I would like to facilitate closing the gap between theoretical population ecology and conservation science by encouraging the use of transient analysis, and further by investigating mechanisms that render populations and communities resilient. And with that, I would like to thank you for your attention. Again, hundreds of field ecologists that are putting their effort into collecting long-term data. I would like to thank Straight Change participants and Wilder team, as well as Department of Ecological Dynamics at the ISW. And last, I will take a moment to show you this slide that I'm including at the end of each talk I give since the 21st February of 2022. I would like to thank you all for standing with Ukraine in the war imposed on us by Russian Federation. And I'm showing this slide not as a researcher, but as a human, as Ukrainian who was born, grew up and studied in Ukraine. And even though I live abroad since quite a while and my small family, my kids and my husband is here in Germany, still my parents, my sister and her family, many of my cousins, their families, many of my friends live in Ukraine. Several people who are very dear to me died because of this war, because of this cruel and unjustified and unnecessary war that was imposed on us by Russian Federation. I know there are a lot going on right now in the world. In every country, there are some crises that needs our attention, and they are closer to you probably. But I would like to ask you to please not give up on us and do not forget us, because what's happening now is a true genocide of Ukrainian nation, and sadly, the best people have died already. I would like to thank you also for all your kind of different kinds of support that you provided for Ukrainians and to your governments for the continuous support in this cruel war. Yeah, I'm sorry for bringing this sad slide last, but as I'm saying, it became a very sad tradition since almost 3 years ago. Now I'm back to research and happy to take questions.
Thank you very much, Viktoriia. So I'm taking over because Maria's now hopped on her plane. But yeah, thank you very much. Round of applause, lots of claps coming in. Does anybody have any questions for Viktoriia? So either raise your hand using the Zoom chat, or if you want to unmute yourself and ask your question, please go ahead. I can maybe start with a question. Viktoriia, so where you were showing the slopes in the phenological responses to temperature, it really did seem like it was almost splitting into two groups, the ones that were responding positively, so I guess advancing the date of that phenological event, and the ones that are responding negatively. I wondered what type of phenological events are falling into those, if you sort of grouped those two, which type of phenological events fall into each, and whether it could be something to do with the phenological event going into winter or coming out of winter. So I wondered if you'd looked into that.
Thanks. This is a great question. So first, and that's something that we tried now to also make in the revised manuscript very clear, the fact that we have this dichotomous split, if you will, between the slopes, and we have almost no slopes around zero, is partly the result of using sliding window analysis to detect the signals. So because we use sliding window analysis, we only remain the studies that have a strong signal, right? And therefore we have nothing around zero, which may seem as, sorry, yeah. And then talking about the particular specific phenological traits that are used, we have a few, not so many, but a few autumn events, but then majority of them are spring phenological events, and yeah, none of them really lines up with being either positive or negative. So there is no a strong structure in there.
Okay, thank you very much. Anyone else got more questions? Okay, I can see Kara. Do you want to unmute, Kara, and ask your question?
Yeah, hey Vic, thank you for that. That was super nice talk. Really enjoyed it. And I was curious about this aspect of it too. You hinted that the intra-species variability was quite high and even compared across the different species. Were you able to look into like how that varied with temperature over time? Like, are we having any differences in variation within the species?
So that's something that some people from the Strait Change project now try to look at in more detail. But now within this data set, because we did not particularly design the search for the data that would cover several populations across their species range, we can't really properly address this question. But yeah, that would be a super interesting next question to address. And I know of Liam Bailey's study, I think 2023 or 2022, it was published, where they look across the species range for blue and gray seeds. And this is a published study already, where the phenological sensitivity actually is changing over the latitude for those two species across. So they have, I think, more than 30. Oh, I'm not gonna lie here, but there are several dozens of populations that they have in the data set, and they do find differential response among populations.
Oh, thank you.
John?
Hi, thank you for the talk. I was just wondering about the time varying stuff for the hyena that you presented. In the figure where you had all the resilience metrics changing through time for the two populations, there's this, I think it was 2011 or something, there's like a dip for the Ngorongoro populations, and well, I guess for Serengeti as well at different times. Do you know what is causing those like shifts, year by year shifts? Like, because I guess that's presumably some response to temperature, and why you don't see that in like the amplification or reactivity so much as you do for the other two. Sorry. Yeah.
Yeah, thanks for this question. So yeah, that's a good question. Why? So basically some of these, and Ella discusses this in her manuscript, that the most pronounced declines that we see, they align quite well with what is known about the disturbances that affected two different populations, and they are different disturbances. So around 2010, 2011, there was a streptococcus infection in Ngorongoro, and that aligns quite well with the dips that you see. But I think your question, and then for Serengeti, there were different disease that affected it at a different time. So many of these dips that are very pronounced, the lows, they correspond to a particular disturbance that took place. About your question, why some metrics are responding more than others, I don't have a good answer. So why we see it on the inertia? Well, the biological interpretation would be that the population size have declined because inertia is affected more than any other metric, right? It would mean that the amplification anyways is the increase. So it increased a bit, it then was mainly the lower the population density that would be found once the population growth achieved what is expected asymptotically was much lower than at the asymptotic population behavior. So it means that this offset, in the end, it was lower. That's why I think maximum amplification is not, would not respond here. So there was not an increase in population size in response to that disease, if that makes sense, I hope. Yeah, then why reactivity does show a decline here, but yes, sure, it's less responding than inertia.
Cool. And do you see similar patterns? Do you see similar patterns when you do the stage biased vectors? So if you, rather than doing the empirical age structure, do you also see those like dips at 2011, for example?
So that's the same that with the stage bias, and I did not explain this in much detail. With stage biased, what we measure the so-called envelope. And so that's why we have upper reactivity and lower reactivity. So we basically measure the envelope of where things can be found. We do not have an exact position. We have a whole range of the possibilities. And this is a bit exemplified. I'm not sure whether you see my mouse because it's, for me, flickering somehow all the time. Do you see it now? Yeah, okay.
Not really.
Yeah. But then if you see on the plot, the black line showing the decline and the gray line showing the amplification. So with the stage biased vectors, we measure the whole envelope. We don't have a one single metric and one single point that can be compared to the time constant approach, if that makes sense.
I see, but it's still time varying. So presumably you still have an annual resilience metric, right?
Yes, we do. Yeah. And that's the differences are the differences in the both. So the MAPE measures the difference. So we do have, okay, maybe going one step back here. We do have this, but it's not that it's value. Yeah, with the empirical structure, we can really put it on the plot if you want. And with the envelopes, it's still a whole range of the possibilities, you see?
Yeah, sure. The kind of thing I'm getting at is just like, I'm just wondering how much of it is driven by the annual age structure shifts and how much of it is, that kind of base scenario where you just do a stage bias vector and like whether you could see a difference in that. But anyway, but yeah.
Yeah, this untangling the effect of initial population structure and matrix in demographic resilience is another part of research that I think Rob Salguero Gomez with his group in Oxford are focusing on now.
Thank you.
Okay, I'll just do one final question from the chat, but then we'll wrap up because we are coming towards the hour. So Jenny's asked, following from John's question, I think there's something I'm not understanding. How do you calculate these metrics from a single year's matrix? do you have to impose a disturbance on the matrix model in a simulation? And if so, what kind of disturbance do you impose?
So the perturbations are perturbations to the demographic structure, and the way we calculate them, they are basically based on the matrix algebra. So many of these metrics that are shown on the list, they are relying on the lambda, the dominant eigenvalue. So they are all derived mainly from the lambda, but then also sometimes the left and right eigenvector comes into the formula. So there are formulas. If you check back Capdevilla's paper, you will see the formula. So they are computed basically. It's not something that we do in a simulation manner, if that helps a bit. But yeah, I'm not sure whether it fully answers the question.
Thank you, Victoria. I think that was really helpful, actually. So thank you again for a brilliant talk.